So I'll tell you a little bit about Arbor Bio. So Arbor was founded about 5 years ago by Fong Zhang and David Walt. So this is the East Coast version of the Doudna version, I guess, of on the CRISPR world. And the view was that Cas9 wasn't the best nuclease chosen. It was actually the first one that worked. And so the thinking was, well, why don't we actually go back and actually take a more strategic review of the entire metagenomic data and identify the best and brightest nucleases, if you will. Can we find nucleases that have differentiated profiles that allow us to have a portfolio view of how to do gene editing? So rather than having a single asset, let's take a portfolio view here and a more traditional approach that you would take with any type of platform technology. So with that, our vision is really to provide curative genetic medicines for all patients with genetic disease. We've built a portfolio in the liver and CNS, and our first IND is targeted for end of 2023. And our differentiation is in a couple areas. So first, obviously, building this toolbox. And importantly, the way we built that toolbox by having the input of folks like David Walt, one of our co-founders, who co-founded Quanterix and Illumina, and brings this high-throughput view of the world, which is really important to allow us to create a platform that allows us to not only screen for interesting CRISPR systems or other systems of interest, but also be able to bring those into the wet lab and do some high-throughput approaches for engineering these nucleases to optimize them for the characteristics we want. We've got a very experienced management team with over 25 INDs and multiple BLAs under our belts across gene therapy, gene editing, and cell therapy, which is very important in this space. And importantly, we've done some partnerships with Vertex I'll talk about. So from a team perspective, we've grown the team significantly over this year. So we brought in John Murphy, who's our CSO, who has significant experience at Pfizer in leading their gene therapy programs. And Pam Stykowitz, who has a lot of experience from Editas in leading some of those gene editing programs. And then Katie McCabe, who's actually was here at the meeting, very well known in our space, and with a lot of experience from both Lilly and Roche. And so with our co-founders, we're able to really create this robust platform that allows us to build this portfolio. And then we've got a leadership team that knows how to translate these discoveries. Now, we see ourselves playing in this next generation genomic editing. I've heard some people call it gene editing 3.0 or 2.0 or whatever 0.0 you want, but it's really the next generation view where we move beyond simple knockdowns that have been the target of the first generation approaches, which have built a great foundation for next generation approaches. And by having this portfolio of bespoke nucleases, it gives us the flexibility on size, on the PAMs or those binding motifs, so that we can target wherever we need to target within the genome. And then utilize other precision genome editing approaches like base editing or prime editing. And importantly for us, our platform, this is not where it stops, and we continue to innovate. So we've discovered CRISPR-associated transposases. So these are can play a role in whole gene editing. So these are transposases that have specificity in bacterial systems, and we're working to translate these into mammalian systems. So it allows us to have this entire portfolio of tools so we can rewrite the genome however we see fit, which allows us to actually start with the underlying disease pathology and then work our way back to what's the right system or approach to take for the patient. I'll talk a little bit about our platform and then where we're going as a company. So importantly, as one of the first movers here, we've been able to look across the entire metagenomic data, both public and private data that we've generated, and aggregated over 3 billion proteins. So we've actually looked at the protein level rather than the DNA level, and that's an important element for actually identifying systems that have interesting profiles for us. And so we've got this cadre of computational biologists that are very talented that can pull out systems of interest. We can then pull these systems into a high-throughput bacterial screening process where we can screen them against 18,000 different guides to really characterize it, to understand, what does the PAM look like? What's the size? What are the other characteristics of this particular system that could be interesting for us? And then pull those into a mammalian engineering system and continue to learn using machine learning and AI approaches with our platform. So today we've got 7 proprietary editors, which gives us a pretty robust toolbox with different PAMs, different sizes, and different other characteristics, including off-target profiles. And it's important, I think you heard a little bit, because it allows us to have different cut types, obviously much smaller sizes, and importantly, that different PAMs allows us to target where we need to in the genome. And this becomes really important when we get to precise genome editing, because you have got to get really close to wherever you need to do whatever edit you're going to do. And if you only have a single nuclease with a very restricted PAM, you're limited. And so that portfolio is important from that perspective. This is a tree of the currently published systems and families. And the blue are the families that Arbor has discovered out there. So many, many families. Each family can be between 5 and 500 nucleases. So you can see there are, probably thousands and thousands, maybe even hundreds of thousands of different nuclease families out there that evolution has created. And by taking this a structured approach, we've been able to build a strong IP portfolio and been able to really identify those editors we want to see, because it's not a stamp collecting exercise. We could collect 3,000 editors if we want to, but to what end? And so having a very strategic approach on how we think about it. The engineering aspects are also important, because oftentimes these nucleases that look really interesting may not work extremely well in a mammalian system. But in this particular case, by putting it through our engineering platform, we can bring those system efficiency up from a very low rate to a very reasonable and high rate that can be utilized for therapeutic approaches. So that's an important distinction for us. Now, over the last few years, we've published in Molecular Cell on our RNA-based nucleases. We published in Science on our DNA nucleases. We've done work on prime-like editing, base editing, and these transposases. So we've built this really strong toolbox of these next-generation editors, which allows us to really have that flexibility to think carefully about where do we want to deploy these and how. So the key, as I said, from a therapeutics perspective is we have to start with the disease pathology. Rather than having a hammer and running around trying to hit everything with it, let's figure out what we're trying to fix first, get to the science, and then work our way backwards. And because we've got this portfolio of approaches, we can really tailor as needed to really impact that particular disease state. Now, there's a lot of areas we could focus on as a company, and we've sort of seen it in 2 big buckets. You've got this engineered ex vivo cell therapy bucket, and you've got in vivo genetic medicines. And on the engineered cell therapy space, the nuclease, it's a key building block, but it's not the therapeutic. And you've heard talks, I think, from different car players and other players in the cell therapy space. And the nuclease is a key part of what they need to build their particular tools. And so this is an area where we capture value through partnerships. We announced a large partnership in this space with Vertex in August, and we'll continue to do partnerships in this space because it's an important way for us to capture value and make sure that folks have access to an unencumbered nuclease to do what they need to do to help patients. And then that allows us as a company to focus in on genetic medicines where we've built our own portfolio, and we can also partner where appropriate. And we actually have another partnership with Vertex in this genetic medicine space. Our portfolio is really focused, as I said, in the liver and CNS. The liver is a fantastic space for us because delivery is, I would say well validated, but it's validated, I think, if you utilize a lipid nanoparticle approach. In addition, it's a space that allows us to demonstrate differentiation from RNA-based or first-generation nucleases, which are just focused on a simple knockdown. And so we take what we call a knockdown plus strategy here. Let's do a knockdown, but let's couple it with an additional type of editing that we can utilize with these next-gen editors. And then in the CNS is obviously an area of high unmet need, but there are so many diseases that we understand the underlying genetics but we haven't been able to have the tools to actually impact those. And so it's an important space for us to think about. Just in the interest of time, I'll skip quickly past that. So where's the company? So to date, we've raised over $85 million, including over $50 million of non-dilutive partnership funding. The company the Series A was funded by Arch. This was about 4 years ago. And then we've done some important partnerships with Vertex, as I mentioned, as well as one with Lonza. The Vertex partnerships, just to talk a little bit about those. So the first partnership, which you see on the left-hand side, yes, your left-hand side, is really focused in vivo editing, where there's 5 indications that Vertex has exclusivity on, and we're working together to really do this strategy of let's tailor it to the underlying disease pathology, bring the right editing approach in for that disease. And then the second Vertex partnership deal, you can see some of the numbers there, was really focused on providing access to our nucleases for 7 indications, or 7 cell therapy programs, including type 1 diabetes, sickle cell, and beta thalassemia. So, Arbor, I think what we've done is we've built a really strong platform for discovery. And we focused initially in the nuclease space and built a really nice suite or portfolio of gene editing technologies that we can utilize today for very specific gene rewriting, and in the future for larger gene rewriting. And so it's a very exciting space. At the same time, we're going to continue to invest in our platform because there's a lot of novel, interesting things that we continue to identify that we can, as we begin to couple nucleases to different types of domains, we can continue to evolve and provide really interesting ways to edit the DNA or the genome. So it's a very exciting time, I think, in this space. There's a lot of activity, and we hope to enter the clinic in a couple years and really see what how well these editors work as we begin to really test the bounds of genome editing. So with that, I'll end and say thank you very much.